Good morning, everyone. Today we will start with the chapter 1. It is the first chapter in our book. It is about basic mechanisms of sexual reproduction. We all know that reproduction is two types, sexual and asexual. Sexual is the mating between two individuals of opposite sexes, but asexual, it is amitotic cell division where the offsprings are identical to their parents. Now we will start with document 3, meiosis. There are two types of cells, diploid and haploid. Diploid have two N chromosomes, which means 46 or 23 pairs, and from the mother or 23 from the mother and 23 from the father. They are all body cells except gametes. Haploid cells have N chromosomes or 23. They are one set of chromosomes and either from the mother, maternal origin, or from the father, paternal origin. They are gametes or sex cells. Chromosomes are classified into two types, autosomes and chronosomes. Autosomes, they are all chromosomes except sex chromosomes, and they are 22 pairs in diploid cells and 22 in haploid cells. While chromosomes or sex chromosomes, they are XX in female diploid cell and XY in male diploid cell, except in poultry they are the opposite, and the female gamete ovum contains the sex chromosome X, while the male gamete sperm contains either X or Y. Chromosomes are condensed form of chromatin. They are made up of DNA or deoxyribonucleic acid, and they are made up of one chromatid, simple chromosome, or made up of two sister chromatids. They are duplicated chromosome, and the two sister chromatids are identical to each other. Homologous chromosomes have same size, position of centromere, and same bending pattern, but they do not necessarily have the same alleles. This is a review about mitosis. Mitosis is a cellular division during which a mother cell with two N chromosomes, which means 46, divides to give two daughter cells, each with two N chromosomes, identical to each other and to their mother cell. Before mitosis, there is interphase during which DNA replicates. Then the four phases of mitosis are prophase, metaphase, anaphase, and telophase. During prophase, the nuclear membrane disappears, the nucleolus disappear, and the spindle fiber or achromatic spindle appears between the two centrosomes, which change into asters, and the chromatin condense into chromosome, which become short and with two chromatids each. Metaphase, where the chromosomes align or arrange at the equator, forming the equatorial plate at the center of the cell. And then anaphase, during which there is polar ascension. Every two sister chromatids separate when the centromere divides and migrate to the opposite poles. Then at the end, telophase, where there is constriction at the middle of the cell, which divides it into two cells. The nuclear membrane and the nucleolus reappear, and the chromosomes uncoil into chromatin, which means they will become thin, and as there's a change into centrosomes, the spindle fiber disappears. Now we will start with meiosis. It is a type of cell division in sexually reproducing organisms. It results in four daughter cells called gametes, each of which has half the number of chromosomes as compared to the original diploid parent cell, which means that unlike mitosis, the daughter cells are not identical to each other and to their mother cell. Here they are different. And then these gametes or haploid cells, which means they have 23 chromosomes each, they undergo union by a process called fertilization, giving a new generation of diploid organisms. And meiosis occurs in the germ cells of sexually reproducing organisms, and here they are the gonads, which is the testis in the male and the ovaries in the female. And meiosis has two divisions. We will talk about them in the next slide. Now we will talk about the two divisions of meiosis, meiosis 1 and meiosis 2. Meiosis 1, it is reductional division where the number of chromosomes is reduced from 2n to n per cell, and meiosis 2 is equational division where the number of chromosomes remains n chromosome per cell. First, here these figures from 1 to 6 show meiosis 1 and from 7 to 9 show meiosis 2. Starting first with meiosis 1, we have prophase 1. The first three figures refer to prophase 1, 2, and 3. First, in prophase 1, the nucleolus and nuclear membrane disappear, centrosome change into asters, spindle fiber appears between the two asters, and the chromatin condenses into chromosome. It means each with two chromatids, as we can see in the figure. Then there is pairing up of homologous chromosomes, as you can see in figure 2. Every two chromosomes that are the same or similar, having the same size and the same position of centromere and same binding pattern, they come close to each other. Then the homologous chromosomes, as in figure 3, synapse or come over each other, forming tetrads where DNA fragments are exchanged. And here tetrads means 4 chromatids. We can see that each two homologous chromosomes come over each other, and they synapse and exchange parts of their DNA. And then we have in figure 4 metaphase 1. It is the lining up of homologous chromosomes at the equatorial plate. And then anaphase 1, as you can see in figure 5, homologous chromosomes separate and move to the opposite poles, and here not sister chromatids. You should pay attention that in anaphase 1 there is separation of homologous chromosomes to the opposite poles. It is called polar ascension. Then we have telophase 1, where first cytokinesis occur or constriction. It is called a cleavage furrow at the middle of the cell, dividing the cell into two daughter cells, each having n chromosomes and each chromosome with two chromatids. Thus we have two daughter cells having n chromosomes, like here 3 chromosomes, half the number, which is 6 at the beginning, with two chromatids each. Then meiosis 2 starts, and it is in figure 7. We start directly by metaphase 2, whereas there is lining of the chromosomes at equatorial plate, and every chromosome is made up of 2 chromatids, and we are working with 2 daughter cells. Then in figure 8, which is anaphase 2, there is the separation of sister chromatids to the opposite poles in each cell, and the centromere divides in the middle, and we have the separation of sister chromatids. Then the last phase, which is the telophase 2, where the second cytokinesis occur to divide every cell into 2 cells, each having n chromosomes and each chromosome with 1 chromatid. Thus we have 4 daughter cells, each having n chromosomes, which here is in this example it is 3, with 1 chromatid each, and the nucleolus and the nuclear membrane reappear, asters change into centrosome and spindle fiber disappear, chromosomes uncoil into chromatin, and these 4 daughter cells are called gametes, and they are not identical to each other and to their mother cell. Now we will talk about the changes of nuclear DNA concentration during the cell cycle. The cell cycle consists of interphase and mitosis, or interphase and meiosis. Now we will talk about the cell cycle and mitosis and the changes in the DNA concentration. C is the concentration and 2C is double the concentration. First, G1, it is a growth phase, and here the quantity of genetic material remains constant. Every chromosome is made up of 1 chromatid. Then synthesis phase, or S phase, where the genetic material becomes doubled, and every chromosome is composed of 2 sister chromatids. That's why the quantity of DNA becomes 2C. Then G2, it is a phase where the cell is preparing for mitosis, and the genetic material remains constant. Every chromosome is made up of 2 chromatids. Then mitosis starts with the prophase, where the quantity remains 2C. Then anaphase, as we can see that there is a drop in the concentration from 2C to C, because there is the separation of the sister chromatids. The chromosome is made up of 1 chromatid, and mitosis will end by telophase. Then G1 start, and a new cycle will start again. Also, in the cell cycle and meiosis, we have the same change, but we have a second division here. It starts first with the G1S and the G2, the same changes, then meiosis 1 and meiosis 2. In meiosis 1, we can see the drop from 2C to C because there is anaphase 1, separation of homologous chromosomes, and then in meiosis 2, we have the second division where we have a drop from C to half C, which is anaphase 2, separation of the sister chromatids in the two daughter cells, and then a new cycle will start again.